I see trees of green, red roses too. I see them blue.
Welcome to Childhood Cancer Talk Radio, powered by Jack's Angels, where we talk about your experiences, advocacy news, research, and today's heroes for kids. We are here to bring hope to the unthinkable but real experience of a child diagnosed with cancer. Jack's Angels advocacy mission is to provide a voice for children with cancer, to effect change for a more hopeful future for those to come. Childhood Cancer Talk Radio starts right now. Here is your host, Janet Demeter.
Welcome to Childhood Cancer Talk Radio, where kids come first for the cure. I'm Janet Demeter, your host, and we have a great show for you today with guests Rhodemann and Clayton Larsen of Vesselon Incorporated, an emerging medical technology company developing a potential solution to one of the greatest challenges to treating pediatric brain cancer, the leading cause of death in children with cancer. If this is your first time listening to Childhood Cancer Talk Radio, we do our best to provide a platform from which children with cancer and their families, researchers and doctors, caregivers and advocates alike can have a stronger voice in the world at large. We hope also to educate and foster connections in our community and inform of new developments in research. So the reason we are so excited about today's guests and the information they bring us is that the landscape of the medical research industry has been relatively bleak for pediatric brain cancer for the last 40 years. There have been literally no new solutions developed in treating the disease that could be translated into standard treatment protocol in all of this time. Because of personal experience, I often refer to a brain cancer called DIPG, diffuse intrinsic pontine glioma, an invariably terminal brain cancer for which parents have been expected to witness an horrible death for their children while nothing has been done significantly to save them in all of this time. And I mention it because it's the second most common pediatric brain cancer, which is unfortunately the leading cause of death in kids with cancer. And it's an extreme example of the difficulty in treatment of pediatric brain cancer and poses two striking challenges to treatment. A, getting medicine to the cancer, as systemic chemotherapy has been dismally unsuccessful in reaching the target due to the blood-brain barrier. And B, the other most significant challenge is the fact that this aggressive cancer is one of the least responsive to chemotherapy, so finding the right medication for treatment of the individual child, even now with experimental direct access of the tumor using microcatheter surgical insertion, can be hit and miss. Such surgeries still in the initial stages of development themselves are quite risky, and not all children survive the surgery insertion of the microcatheters for actual treatment. And so for brain cancer, these two strong challenges have remained nearly insurmountable for decades. Even so, development of the direct access with microcatheter or catheter insertion for treatment is progressing, showing definite promise. Families are petitioning for the experimental procedure on a regular basis in the UK and here in the United States with Dr. Suidan of Sloan Kettering in New York. Yet the risk from both fronts is still undeniable. So this show is dedicated to the memory of Parker Landis and his family. His father joined us on a show last year entitled The Realities of DIPG, and his doctor, Mark Suidan, joined us on yet another episode to describe his mission of finding solutions for this dreaded disease using this catheter insertion for treatment. I'm very sad to report that Parker passed away just last week, succumbing to complications from DIPG. But according to our guests today, 99% of existing neurological chemotherapy and therapeutic drugs cannot reach their target in the brain effectively. Rhodemann and Clayton Larsen are partners in the company Vesselon, which has a non-invasive ultrasound and microbubble technology platform that will enable small or large drug molecules to cross the blood-brain barrier, or BBB, safely by transiently opening this barrier. This method is intended to enhance the delivery of drugs, improve treatments for brain cancer, Alzheimer's, Parkinson's, and other neurological and central nervous system diseases without having to change the drug molecules in any way. The company is founded on the confidence that the combination of this ultrasound technology and FDA-approved microbubble, acquired in 2016, provides them the unique opportunity to share the technology with the world in commercial development. Mr. Lee is the founder of Vesselon, is a serial medical device entrepreneur, and brings more than 30 years of experience in finance, business development, strategy, patents, and regulatory submissions. Mr. Lee holds a BA from Middlebury College and an MBA from the University of Chicago, and there's a world of experience in between there. Mr. Larsen co-founded Vesselon in 2012 and brings over 30 years of executive management, marketing, sales, clinical innovation, and engineering project management in the healthcare industry. Mr. Larsen holds a BS in molecular biophysics and biochemistry from Yale University. Gentlemen, welcome to Childhood Cancer Talk Radio. We are so grateful to have you with us today.
Thank you for the opportunity.
Good to be with you, Janet.
This is great. So, wow, could you tell us a little bit about how you came together with this idea and how Vesselon came about?
Sure, Janet. This is Rhodemann. Basically, we are we're healthcare professionals that are very interested in finding solutions to unmet needs, and the company came about back in 2012 when I was looking around for some technology to commercialize and focused specifically on medical devices, medical technology. And having previously founded or co-founded an orthopedic implant company, I kind of cut my teeth on the field and really fell in love with medical technology and the ability to help people. And so after contacting numerous academic institutions and looking on their technology transfer websites, came across a very interesting technology that was the basis of Vesselon, which was the use of ultrasound, non-invasive use of ultrasound and microbubbles, actually to help treat stroke patients in a pre-hospital setting. And so doing a little bit of due diligence about that technology, I began to pull together a team and came across Clay through a mutual friend. And with Clay's background in senior management as well as in ultrasound, we joined forces and pulled together a business plan and have started product development on that product. And then, as you mentioned a little while ago last year, we acquired an FDA-approved microbubble. And once we acquired that microbubble, we began looking around, now that we control both ultrasound as well as the microbubble components, we started looking around for additional applications and technologies and came across a very interesting work that Dr. Elisa Konofagu at Columbia University had been doing for the past 10 years to open up the blood-brain barrier and to enhance drug delivery. So that's sort of the genesis of Vesselon.
Could I ask you a question about the microbubble?
Absolutely.
How it's FDA? How do you approve? How is the microbubble? How was that approved? It just it sounds like it must have been. What was the process like?
Well, why don't you take that, Clay?
Yeah, Janet, this is Clay. Microbubbles are actually part of a class of things used in diagnostic imaging called contrast agents. If anybody has an X-ray or a CT or an MR exam, they use different types of contrast agents depending on that imaging modality, as they call it. Contrast agents have been around 30 years now to help ultrasound highlight vasculature, and they are primarily used for cardiology to help view the left ventricle better. So that's what these microbubbles are for, because they're about half the size of a red blood cell, and so they course throughout the body wherever the blood goes, even into the finest capillaries. But they were approved by the FDA specifically on a diagnostic basis for imaging the heart. So this is now the basis that people have done research to show that there's a myriad other applications for these microbubbles that really are quite amazing. One that Rhodemann mentioned that we initially got into was the ability for ultrasound and microbubbles to dissolve clots that cause stroke. But then there's also this fantastic mechanism, whereas the microbubbles inside small capillaries, when hit with the right frequency and energy of ultrasound, can actually relax the blood vessels to temporarily and safely open the blood-brain barrier.
That's amazing, and thank you for describing it, because I'm not a medical or technical professional, and it sounded funny, and I just thought, well, that other folks listening might really benefit from hearing your description as well, because it's fascinating. And one of my first thoughts is, well, are they microbubbles? Do they contain air? Do they contain medicine? How does that work?
Well, let me take that too, Janet. With the microbubbles actually have an inert gas in the center, and then they have these little very thin flexible shells made out of phospholipids. And phospholipids, not to get technical, it's actually what the cell membranes in our body are made out of. So you have these little tiny microbubbles that travel throughout the bloodstream. They do not chemically react with anything. They're not involved with the actual, they just act like almost like little tiny mechanisms inside the blood when they are hit with ultrasound. They basically oscillate, and it's the oscillation, which the technical term is called cavitation, that actually is effective, and it's the mechanism by which all these things happen inside the bloodstream. And it's been an extremely well-tested technology. To give you an idea, when we say FDA approved, the contrast agents, the microbubbles are considered a drug by the FDA. So they've gone through rigorous testing, literally dozens and dozens and dozens of tests, both in animals and humans, with a full safety profile for them.
Incredible. Well, let's come right back. We're going to take a quick break. We'll be right back after these messages from TogiNet.
Stay tuned. Janet will be right back with more on Childhood Cancer Talk Radio, where kids come first for the cure.
It's the Fitness Minute with fitness expert Annette Hammond.
To lose weight, we know that each day we need to burn more calories than we take in through eating, and exercise burns more calories. According to Discovery Health, a 150-pound person will burn about 60 calories while taking a 1-hour nap. 1 hour of sitting and watching television burns about the same. But if that 150-pound person takes a 1-hour brisk walk, then say goodbye to more than 250 calories. Cardio exercise like running, biking, swimming, and brisk walking are the best modes of exercise to burn the highest amount of calories and will get the endorphins flowing in your body. Those feel-good neurotransmitters boost your mood naturally. So use exercise to burn calories, lose weight, and to feel good. I'm Annette Hammond. To hear other fitness and weight loss tips, visit our website at AnnetteHammond.com.
It's words you never heard.
Derek Thompson, co-founder of the Save the Elephant Foundation, was taking a rescued elephant named Kamla for a walk along the river. As they were walking, Derek decided to stop and take a dip in the water. But when he called to Kamla, who was waiting on the shore, the elephant thought he was in trouble and came running to his rescue. Kamla charged through the water, sheltering him with her body and offering her trunk for him to hold on to. What's another word for a trained elephant? A kunki. Elephant trainers in Asia are known as mahouts. And the padded seat or saddle used to ride on an elephant is called a howdah. Kamla and her mother, Bai Tui, are among the 70 elephants the Foundation has rescued since its inception.
It's words you never heard.
I'm Carolyn Davidson, and you can have fun challenging your words you never heard vocabulary with my free app, Too Funny for Words.
You'll always have my shoulder when you cry.
Welcome back to more great conversation on Childhood Cancer Talk Radio, because every child's life deserves hope. Here's Janet.
And we're back with Childhood Cancer Talk Radio on TogiNet.com, powered by Jack's Angels Foundation for Pediatric Brain Cancer Awareness and Research and Advocacy for Children with Cancer, on the web at www.jacksangels.org. Our distinguished guests today, Rhodemann Lee and Clay Larsen, are co-founders of Vesselon, a company dedicated to bringing urgently needed technology for treatment to the field of neurology, and most important to our mission here at Childhood Cancer Talk Radio, pediatric brain cancer, the number one killer of our children with cancer. Pediatric brain cancer poses two challenges to treatment, two major ones anyway: accessing the tumor and developing an effective chemotherapy treatment. Number one is access, for we cannot progress in our discovering the right treatment without safe and direct delivery of medication, and Vesselon's microbubble and ultrasound technology platform offers real solutions to this problem. Gentlemen, before the break we were discussing the logistics of these microbubbles. I had questions about it, and you were describing the approval process with the FDA and how that came about, and I had to cut you off because we went to break, so I was hoping we could pick that up again.
Yeah, I think the most important thing, Janet, is that the components that we're using, both the microbubbles and the ultrasound, individually are extremely safe, and they're extremely well known by the FDA. And so it's actually the combination of them when they're used together. That's when you have some very interesting effects happen, where when you get these bubbles into these small capillaries, and when they oscillate at that right amount of ultrasound energy, it basically relaxes these tight junctions that are in the vessel walls that actually protect the brain. It's really the brain's natural defense mechanism, but the brain's natural defense mechanism also defends it against things like chemotherapy that's meant to try to treat these tumors.
Right. So when they're having this treatment and the ultrasound is applied, so the relaxation of it, so to speak, would only occur at that time. So it's correct. Is that correct? So that it would, I could see all the more the fact that it's, safe, because usually when there are treatment modalities and they alter something in the body so that it will allow medication, usually it's not necessarily a start and stop thing, or it's a general condition that a person has to endure that could invite other complications. And this sounds like it's sort of like an on-the-spot treatment. Is that true?
Exactly. For instance, the barrier only opens up where the ultrasound focused, and so we can target that right at the area of the tumor. For instance, you mentioned the catheter-based technologies,
yes,
which have to essentially traverse physically, has to go through the entire brain to go ahead and inject. It's called convection enhanced delivery, whereas we can open up the blood-brain barrier so that the chemotherapy, whether it's an infusion or whether it's a tablet, an oral medicine, as each time it passes by the tumor, then the medicine that's in the blood will then be absorbed directly in only at that spot where the tumor is at the focus of the ultrasound, and it would only be temporary. It closes up within less than a day.
Wow, that's fascinating. Wow. So what sort of assembly of scientists did you have together to work on this, or to put it together for Vesselon? Are there collaborating scientists that you have that that are helping develop the technology further?
Yeah, let me take a shot at that, Janet.
Sure.
It's Rhodemann again. We are very much focused on commercializing products, having experience in medical technology and healthcare. And so what we wanted to do was identify groundbreaking technologies that have been developed at academic institutions. And we, as I mentioned earlier, we're fortunate that we came across the work that Dr. Konofagu has done for the past 10 years or more at Columbia, where she and her group have been able to characterize the ultrasound parameters and microbubble properties that are necessary to really safely open up the blood-brain barrier to allow drugs of various sizes to get across. And so we are working with Dr. Konofagu to commercialize her technology. And she is one of just less than a handful of what we would categorize as world-leading experts in the field of using ultrasound and microbubbles. And we're fortunate to be working with her and her lab. Beyond that, on the clinical side, we are actively engaged in talking to a couple of opinion leaders in the neuro-oncology, particularly pediatric neuro-oncology space, at a couple of comprehensive cancer centers in the U.S. And those discussions are at the sort of early stages, so we can't disclose who those parties are, but as business people focused on commercialization, we recognize that we need to engage key opinion leaders from various areas, not just the technical side, but also the clinical side, and there are going to be all sorts of people that we're going to want to engage in our conversation.
Yes, absolutely. And I have another question that is somewhat technical, I would think. And insofar as it's very hope-inspiring, actually, to hear of the focus on pediatric neuro-oncology, because, as I've stated a few times, and you've stated as well, there have really been so few solutions for these children who regularly are diagnosed with pediatric brain tumors. I have a question about, talking about the blood-brain barrier and how you explained very, very well how the microbubbles can relax the site of where you would like the medication to go through. How does that work with a tumor like, for example, diffuse intrinsic pontine glioma, where it's in the center, it's in the pons area of the brainstem? Do you have an entry point at the edge of the brain and then again at the tumor, or how does that work?
Yeah, Janet,
theoretically.
when you want to access a particular part of the brain, there's two things that you want to consider when you're using ultrasound. One is what's the shortest distance, because that way you have just less tissue that you have to go through before you actually get to the place that you want to treat. But then the other point is that you would also like to be as perpendicular as possible to the skull, so that you can get more of the energy getting through the skull. The more acute angle that you approach the skull, that it's you would have more ultrasound energy, more bouncing off, so to speak, as opposed to going into the skull. So if you look at those two parameters, then for accessing DIPG, and we've given this a lot of thought, you would basically go through one of the sides of the occipital part of the skull, the lower back of the skull, from one of the sides, right in the middle of the occipital bone. It can actually get a little thick, so you'd take one side, and then you have actually a very good, maybe 5 or 6 centimeters of distance to get directly into the tumor.
Interesting. And I appreciate your indulging my curiosity. And I don't know if any of these things have been, if it's purely theoretical, if any of them have been applied in any way in animal studies or anything like that yet. But I appreciate your taking that step further and going, what if, and how would we potentially treat this? Yeah.
But the animal studies have actually demonstrated many of these things. That's why we think what they refer to as translational medicine is so much of these things have already been worked out by both Columbia and other research institutions. And a lot of the work that we're also benefiting from has been towards Alzheimer's research, where they are trying to, for that application, to deliver drugs to what's called the basal ganglia. Again, not to be too technical from an anatomic standpoint. So a lot of experience has already been researched on how to deliver this focused energy deep within the brain. So therefore, the brainstem, which is affected by DIPG, would be at similar depths to what has already been demonstrated in animal models.
Excellent. Okay, so how many, you may have already told me this before, but how long has this been? It's been a few years now that they've been researching this on animals, and I'm sorry, then we'll have to go to a break, but.
Yeah, it's been about a little over 10 years at Columbia.
Okay. Wow. Specifically on the brain.
Correct.
Fantastic. Wow. I appreciate this so much. We're going to take another quick break here on Childhood Cancer Talk Radio. I'm Janet Demeter, your host. I'm speaking today with Rhodemann Lee and Clay Larsen of Vesselon, fantastic company that is bringing this new technology hopefully to market and wider use in our medical research institutions and ultimately to save our kids' lives. We hope it will help in that process. We'll be right back after these messages from our TogiNet sponsors. Don't go away. We'll have more for you when we get back, and advocacy news.
Stay tuned. Janet will be right back with more on Childhood Cancer Talk Radio, where kids come first for the cure.
Secret Cuisines and Sacred Rituals is a quest, a place, and a feast. Join host Vilasi Venkatachalam every week to explore myths, mystique, old medicine, and brilliant modern solutions through a dazzling kaleidoscope of cuisines, cultures, and cures. This is the place where tribes gather, strangers and familiars, to be memory keepers and makers of our evolving, enduring, evergreen, spoken legacy of wisdom and ingenuity. In Vilasi's words, when we do old things in new ways and new things in old ways, we paint with an inspired palette, weave our own healing traditions, and become our own guru. Vilasi is a troubadour of secret cuisines and sacred rituals. She collects stories of wisdom, ingenuity, and grit. She believes wellness and transformation happen when you stand at the threshold of delight and discovery. She displays her hidden penchant for drama when she leads the safari at the supper club. Her favorite pastime is to extol the marvels of cuisines, cultures, and cures. To her audience in workplaces, seminars, and salons, her mantra is, be your own guru. She is a biochemist, botanist, and alchemist who likes to churn delightful, useful things from a brew of art and science, ancient and evolving, old medicine, and new cures. Join Vilasi every Friday at 11 a.m. Eastern Standard Time, only here on the Woohoo Radio Network.
It's words you never heard.
Any
emergency room nurse will tell you they receive some weird and wild calls. I used to be the night ER nurse at Hennepin County General Hospital in Minneapolis, and remember getting a call from someone who was worried about if it mattered or not if their belly button was an innie or an outie. It doesn't matter, but if you're an outie, you are definitely the minority. Still, that's good news, because you don't have to worry about a little problem that people with innies have. Pledge it. That's another word for belly button lint. Researchers say the color of the pledget in your belly button is related to the clothing you wear, just like the lint that collects in your clothes dryer. A friend of mine from the Philippines says they call it a chikachu. What do you call a person who's obsessed with their belly button? An omphalopsicite. I'm Carolyn Davidson, and you can have fun challenging your words you never heard vocabulary with my free app, Too Funny for Words.
Welcome back to more great conversation on Childhood Cancer Talk Radio, because every child's life deserves hope. Here's Janet.
And we're back with Childhood Cancer Talk Radio, where every child is a life, not a number, here on TogiNet.com. I'm Janet Demeter, your host, and before we return to our conversation with Rhodemann Lee and Clay Larsen of Vesselon, we have this brief bit of advocacy news in childhood cancer. And I have to add to all this that, now's a really important time to advocate. I know that our political climate is somewhat crazy and uncertain. We need to, all the more reason to move forward with our agenda of advocating for children with cancer, that they have more attention and we find solutions for them more quickly. Here's this brief bit of news. This just in on Monday, which was the 27th of February, in Washington, U.S. Senators Marco Rubio of Florida and Michael Bennet of Colorado, Crispin Holland of Maryland, and Cory Gardner also of Colorado introduced the Race for Children Act, Research to Accelerate Cures and Equity for Children Act, legislation supporting the development of innovative and promising cancer drugs for children. U.S. Representatives Michael McCall, G.K. Butterfield, Sean Duffy, and Yvette Clarke introduced the companion bill in the House. And he had this to say about it: pediatric cancer impacts too many families in Florida and across the nation, including my own, said Rubio. I'm proud to reintroduce the Race for Children Act, which would encourage more treatment options for children battling against cancer. This bill takes a further step in extending medical advances in adult cancer treatment to children fighting this horrific disease. I'm especially honored to reintroduce this bill on behalf of the many advocates who have relentlessly fought to get more attention, funding, and resources for childhood cancer. I look forward to continuing my work with them to make this crucial legislation a law. Well, there's Marco Rubio. He's a go-to guy now for advocacy. In Colorado and around the country, researchers are making dramatic advances to treat and cure cancer, but there's even more we can do for our kids, said Bennett. He's the senator from Colorado. This legislation is a necessary update to our laws and has the potential to save thousands of children's lives. I look forward to continue working with parents, researchers, and other advocates to advance the bill to expand treatment options for children. The bipartisan bill, first introduced to the 114th Congress, would update the Pediatric Research Equity Act, or PREA, to reflect the latest advances in cancer drugs. PREA was enacted by Congress in 2003 to address the scarcity of information about how to treat children with drugs developed for adults, and therefore develop pediatric data during drug development. Although PREA has resulted in new information on how to treat children for a multitude of drugs over the years, there are still limited pediatric studies for cancer drugs. And then we also have the STAR Act, which was introduced to this new Congress, the 115th Congress, on February 3rd. In the House of Representatives, February 2nd in the Senate. Designation HR 820 and S 292 respectively. This bipartisan bill is designed to advance pediatric cancer research and child-focused cancer treatment while also improving childhood cancer surveillance, not the French pronunciation, and providing enhanced resources for survivors and those impacted by kids' cancers. The Childhood Cancer STAR Act of 2015 was passed by the U.S. House of Representatives, but did not clear the Senate before the end of the session this past December 2016. The acronym STAR stands for Survivorship, Treatment, Access, and Recovery, that each one of these crucial issues affecting children be addressed proactively with the support of law. So definitely, when you call your congressional representatives, please request that they sponsor this, and quickly. For more information on this and all important childhood cancer legislation that's currently needing support, please go to kidsvcancer.org. Another good one is the Children's Cause for Cancer Advocacy at childrenscause.org, and we will do our best at jacksangels.org to keep you updated on those things. Next on the list is House Resolution HRES 69, the National DIPG Awareness Resolution, which designates May 17th during Brain Tumor Awareness Month as National DIPG Awareness Day. There's been literally no change in standard treatment protocol, radiation, steroids, palliative care, and hospice, nor in its terminal prognosis for over 50 years. Neil Armstrong's daughter died of it in 1962, and there were many cases, high-profile cases even in the news just in the last 2 years, but you'd never know what it was. Always referred to as a very rare terminal brain cancer in the media, DIPG is the second most common pediatric brain cancer and is responsible for the most pediatric brain tumor deaths annually, affecting some 200 to 400 children regularly each year in the United States. The resolution asks for this awareness day and makes the plea, and this is important because this is important for childhood cancer. It using DIPs as a powerful example. It makes the plea for greater consideration for children and for high mortality rate cancers with our government funds, your tax dollars and mine, purposed for cancer research, and in the private sector alike. Please support this urgently needed resolution for greater public awareness and ultimately more research funding for a cure from our government. Its designation is HRES 69. May 17th is not that far away. Please encourage friends and family across the country to ask their reps to sign it. Using the powerful example of DIPG, again, the relative neglect of pediatric cancer research is underlined and is an attempt to have this smaller DIPG community better known. Currently, parents must be resigned to the fact that there are no solutions for their child, and with this awful acceptance, must endure watching their young child die without exception for decades now. You can support action days for it and run or walk with me and Beth Ann Telford, now a veteran of the World Marathon Challenge, on or around the 17th of each month through May 17th, and post your pictures to Facebook to support. It's called the Virtual Relay for Jack, with open registration right now through April 30th, and you can walk or run the event from virtually anywhere in the world. Proceeds benefit a research project for DIPG at Children's Cancer Therapy Development Institute. Visit curedipg.info to register, and for more information on the virtual event, visit our advocacy page frequently for updates on childhood cancer legislation that needs your support at jacksangels.org. Please help DIPG children, brave warriors in the front of terminal pediatric cancer, a day of national recognition. There's no change without awareness, no hope without spreading the word. Please help us honor those currently fighting to stay alive and the tens of thousands who have bravely faced their untimely death over the last 40 years. Thank you. You can visit hres69.org for more detailed information on our action days. And now returning, I think I don't have anything else that I have to mention here. Oh, just one quick thing is this resolution complements the Across the Map project of the Michael Mosher Defeat DIPG Foundation at defeatdipg.org, which has volunteers working in as many states as possible calling for that May 17th Awareness Day. And with that, I want to mention how important our guests are to us today. They and their company Vesselon represent a potential solution to one of the greatest challenges of treating pediatric brain cancer, which is the leading cause of death in children with cancer, unfortunately, and in urgent need of solutions. I'm talking today with Rhodemann Lee and Clay Larsen of Vesselon again, and we were discussing before the break the FDA approval of these microbubbles enhanced with ultrasound technology. in combination, that this is their special patented technology, and it has multiple applications in neuro-oncology and neurology. In fact, I believe we were talking before that there's been some 10 years of animal study. There's been study on how it affects Alzheimer's and other neurological conditions. If you could just quickly for me kind of recap all the different applications that this might presently foreshadow having.
Sure. This is Rhodemann again. So as you pointed out, this is a technology that we believe has very broad applications, specifically in neurology or in the brain, but also the ability to enable drugs to cross membranes is not limited to just the brain. It's just that we're focusing on that just because of our own plan. But just within the brain, we are targeting pediatric malignant brain tumors as our initial application of the technology. And then also, or beyond that, we would target adult brain cancer, and then perhaps Parkinson's, Alzheimer's, Huntington's disease, and other rare and orphan diseases as well, focusing on the brain. But then outside of the brain, one could envision using the same technology or variations to our device to treat other solid tumors or other areas of the body. So it really is a very broad platform.
That's fantastic. And especially because you are focused on, at this time, helping the field of neuro-oncology, because it's in, any disorders of the brain are still quite mysterious to us and to our science. And I think, thank you for bringing this application to such an area that urgently needs it. So what, let's see, we're almost time for a break, but before we go to break, just what preclinical work has been done with this particular technology at this time?
Yeah, one thing that maybe we can continue after the break and actually talk about the clinical work that's already been done,
Yes.
with real patients. But when you look at medical research, typically what they try to do, Janet, is they try to find the right animal model for the particular type of disease that they want to go ahead and investigate. And it turns out that in the brain, the two types of animal models would be the mouse animal model, or the rodent, because it can also be mice and rats, but then also using actually monkeys, small rhesus monkeys.
And let's come back to that. We're going to take a quick break, and let's come back to the mice and the monkeys after these messages from our TogiNet sponsors.
Stay tuned. Janet will be right back with more on Childhood Cancer Talk Radio, where kids come first for the cure.
It's the Fitness Minute with fitness expert Annette Hammond.
Did you know that the average teenager drinks twice as much soda as milk? Since 1983, sugar consumption in the U.S. is up 28%. Why is that? There are several reasons, but one of the most common is soft drinks. 20-ounce beverages have become the norm, and it's not surprising to find that 43% of our sugar comes from drinks. Sugar is blamed for poor nutritional diets. USDA data shows that people whose diets are high in added sugar eat less calcium, fiber, iron, protein, and many other important nutrients. Fat-free foods are also a culprit. Since sugar is fat-free, many people tend to think it's okay to eat as much as they want. Remember that just because a food is fat-free does not mean that it's calorie-free also. For the Fitness Minute, I'm Annette Hammond.
It's words you never heard.
When breast residents of Alligator Point, Florida, discovered a 400-pound gaberlunzie bear raiding their garbage, they called the local wildlife authorities. The officers came out and shot the bear with a tranquilizer dart in order to move him. Unexpectedly, the frightened bear swam out into the water, where it started to drown as the tranquilizer began taking effect. Adam Warwick, an officer from the Wildlife Commission, jumped into action, swimming towards a juggernaut bear, while the other officers tried to figure out how to rescue both of them. Adam was somehow able to grab the bear and paddle 25 yards to the shore, saving the bear's life. The bear was then loaded on a truck and transported back to its home in the forest. What's a word for a last-minute attempt to get something done? A charrette.
It's words you never heard.
I'm Carolyn Davidson, and you can have fun challenging your words you never heard vocabulary with my free app, Too Funny for Words.
You'll always have my shoulder when you cry.
Welcome back to more great conversation on Childhood Cancer Talk Radio, because every child's life deserves hope. Here's Janet.
And we're back to Childhood Cancer Talk Radio, where kids come first for the cure. I'm Janet Demeter, your host. I'm here with Rhodemann Lee and Clay Larsen with Vesselon, and we were talking before the break about in getting eventually here to the exciting things that are happening clinically, the preclinical work that was done in developing this treatment. And we were talking before about using that the mouse model and the monkey model were especially helpful.
Yes, and where the mouse model is very helpful is where they can actually implant different types of tumors and test different types of drugs. And that's exactly why they are actually very hopeful that the drug panobinostat may be helpful for DIPG. Because there's some evidence when you look in the mouse model. Now, when you use monkeys, typically they're strictly for safety and behavioral testing. So they're typically not implanting tumors into the monkeys. But what they do is they'll do a full safety boundaries of the technology to make sure that the ultrasound and the microbubbles, that they know exactly the right settings, the right dosages, and that it does not actually impair any either cognitive or behavioral function. In fact, some of the tests actually show that in certain reaction time tests, the reaction time actually got better when they were using ultrasound and microbubbles.
Oh, amazing. So our monkeys and mice are heroes then, doing some pre-testing for us.
No question about it.
And so moving forward into actual clinical testing, what has been done so far?
Yeah, there's actually been a couple of approaches using ultrasound and microbubbles in real patients that have glioblastomas, adults.
Oh, wow.
One technique is a surgical technique that's being tested in France, where they actually have a burr hole, which is really a hole that's drilled in the skull, and then placing an ultrasound transducer in that hole, using ultrasound and microbubbles to treat tumors, and that's been shown to be have some indication of benefit. It was a safety study. That was the primary purpose of it, and it's been shown to actually be very safe, other than it's a highly invasive surgical procedure. And then in Canada, they have used some very large equipment called high-intensity focused ultrasound. It actually needs a dedicated MR scanner to guide it, but they have done some adult glioblastoma patients and successfully shown how they can open up the blood-brain barrier to then deliver drugs across it. The particular approach that we're working with Columbia to commercialize is actually a handheld device that will be much more practical to be able to be used in an outpatient setting wherever patients are getting chemotherapy.
And that's really cool, actually. Sort of like
Janet,
yeah.
Sorry, if I could just add to,
Yeah, please.
With respect to our approach and the Columbia technology in particular, the process to open up the blood-brain barrier using ultrasound and microbubbles, it takes all of 30 seconds to a minute of ultrasound energy. And as Clay mentioned earlier, the blood-brain barrier stays open for a matter of some number of hours, and we could actually control the duration of the opening. So
that's amazing.
What we believe is that this approach of using a handheld device or a portable device is particularly suited for opening up the blood-brain barrier, and then also to be used for pediatric patients too. So compared to surgery, compared to an MR-guided machine, to us it seems very logical that this is a great application of the technology.
Absolutely. It also would seem that it would help making, perhaps, I know there's a big move right now to go to combination therapy determination, that certainly the process would be maybe a little less dangerous and easier to quickly ascertain, which medications would work best for any individual patient. I know that's an issue because once they go in there with the catheters and implant them, and like what they do in the UK, what they might put in there is it's not a complete crapshoot, but it's scary. It's because it's non-trivial what the result might be. I'm sorry to report that just recently there was a child who didn't even make it past, unfortunately, the surgery. It's a very risky procedure. So this really is just so hopeful. How soon do you think that this might actually be able to be used with decent access for people that want to use it? How long do you see it taking to get to people?
Yeah, I'll take a crack at that as well.
Yeah, children.
Yes. So our plan is to seek accelerated regulatory approval through FDA, and we're actually in discussions with FDA now on the appropriate programs and regulatory pathway. But if we're right that we would be able to focus our indication for pediatric malignant brain tumor patients, then if FDA were to agree, then we would be able to benefit from accelerated regulatory approvals, and then we would go through the process of enrolling patients and conducting our safety study and hopefully obtaining FDA approval in a short order. So until we go through that process, it's really hard to tell. But based on the strength of the preclinical data, the safety profile of ultrasound and microbubbles, we're making every step that we can to accelerate not only the development but also the regulatory approval, and to get this in the hands of clinicians. To your point, Janet, to be able to allow them to try different combinations for patients, or personalized treatment for the patients.
So, yeah, because sometimes, isn't it true that certain technologies often are introduced with a certain drug, and they're kind of limited that way? Is that? Do you think that might change in the future somewhat?
Well, that's actually the beauty of of our approach, in that we're drug agnostic, and we would allow the clinician to make the decision of what drug to try for that patient.
Fantastic.
And there's different types of therapies. People may have heard some of the terms. Obviously, chemotherapy is one that everybody's heard of, but you also have immunotherapy, where they're using monoclonal antibodies. Other diseases need enzyme replacement therapy. And then you also hear of gene therapy, especially in cancer. And if you kind of look at each of those therapies, the size of the molecules kind of increases. And what's exciting, Janet, is all of the research up till now has definitively shown, it's not even a question, definitively shown that this approach can actually deliver even the largest what they call gene vectors into the brain. And so that's what's very exciting. What Rhodemann is saying is that any particular avenue that a clinician wants to take with a particular disease type, we would be able to take their chosen medicine, and deliver that molecule successfully right at the selected target.
That's huge, because that goes right along with all the new things we're trying to do with precision medicine. And I know oftentimes with brain tumors now they're starting to biopsy some of them that hadn't been so forthcoming before, and really creating medicine specific to the child that have to do with their particular genetic makeup. And so that's music to everyone's ears, I'm sure.
Yeah, and one more point, Janet, if I may. Just sort of talking about the applications of this platform technology. We've obviously spent a lot of time talking about the chemotherapy aspect of it, or the benefit of our technology. But what's also been shown in the preclinical model is that using ultrasound and microbubbles can actually enhance radiation therapy and convection-enhanced delivery of drugs. So it's really and we talked about it before, it's really a platform, but there's so many different applications and so many different ways that could potentially help patients. It's
amazing. Well, this is also, in my opinion, a testament to how those of you who work so hard and have done so much research in bringing technologies to market, that is such an important aspect of this whole game of our medical research industry. And I applaud you for what you're doing, because you have a humanitarian focus for not that other companies don't, but I'm just saying it's nice when that's so obviously, your primary consideration is getting this to patients, and that's what it's all about. And I can't thank you enough for what you're doing. I want to let people know where they can follow your progress with it at Vesselon.com. Is that right?
That's right.
They're going to cut us off in a couple minutes here, but it's at VESSELON.com. I'm sure there will be updates there on in the future of what's happening with the technology. But to Rhodemann Lee and Clay Larsen, thank you so much for coming on today and sharing this with us. I hope that we can call on you in the future to come back.
Absolutely.
It would be our pleasure.
Well, it's been an honor speaking with you today. Okay, folks, this is huge, bringing this technology to help our kids with brain cancer and so many other applications. I'm Janet Demeter with Childhood Cancer Talk Radio. It's been a pleasure speaking with all of you, to our guests today, and we hope that you come back and see us again. Give us some awesome updates, because it's amazing. Thank you.
Thank you.
I see them blue for me and you.
We hope you have found inspiration from today's show. For more about Janet Demeter and her mission, visit jacksangels.org. As founder and president, Janet strives to assure that children with cancer and their parents have a voice.